I'm Daniel Freund from the Technical University in Dresden, Germany. Our lab develops and manufactures ATMPs, that's Advanced Therapy Medicinal Products, in the university hospital. In 2019, we had the idea to replace our clean room with an isolator technology. However, the problem at that time was that we had absolutely no idea on how to work inside such a system. Luckily, SKAN just launched a small, compact version of the so-called pure, which we could use for our first proof of principle study. Since we came from academia, we had a quite limited budget, so the new compact system really fitted into our approach. After we got the first system, we realized that there are quite some limitations we didn't think of at first hand. So you have to keep in mind that the space inside the chamber is quite limited. Compared to the clean room, we have quite a lot of additional transfer steps to get material in and out. So that was quite a challenge at first hand. So that's why we came up with SKAN again to see to which extent we can modify the small system to a larger module to really fit all our needs for the manufacturing and also to completely build the process just in one piece. One of the key part is really now the integrated incubator, because if you want to expand cells under GMP conditions, it's necessary to have the incubator in your working area. The next point is that we now have an RTP port in the chamber, which allows us the rapid transfer of liquids in and out of the system. Then we have newly designed transfer locks, which really allow the seamless passing of products or materials from one end to the other. We also have a fill and finish chamber, which is purely dedicated for cool work. So we have a cooling plate, which allows us to keep the cells constantly at low temperatures. The new system will be used for the whole process of isolating cells from a donor organ, expanding the cells in the incubator, and after 3 to 4 weeks doing the fill and finish to have the patient doses at the end. So it's a real all-in-one solution. That was one of the first ideas why we wanted to switch from the clean room to the isolator, because it has a much lower footprint. You do not need loads of ventilation system and so on. Then the construction and maintenance costs are much lower compared to a classical clean room. And the major contamination risk in GMP work usually is the operator. And since we now have a real great barrier between the operator and the product, this reduces enormously the contamination risk. The first lesson for us was that you cannot really transfer one project from the clean room to the isolator without changes. It's really important to think of the process as such. And finally, don't hesitate to ask the experts to help you with process development, process optimization, to get to a result in less time than usually.
We are very aware of the challenges in the cell and gene market because these therapies are labor-intensive with a lot of manual steps. That's why we developed customized solutions.
I'm really looking forward to see this new pure2 in our lab in Dresden. The success of our cells will tell us the success of the whole project.